Welcome to your first semester of cell biology at Delhi University. Today, we are going to explore a fascinating topic: secondary messengers. Have you ever wondered how a tiny drop of adrenaline can make your entire body react in a split second? The secret lies in how cells communicate internally. Let us dive in and uncover the hidden communication networks inside our cells. To understand secondary messengers, imagine a massive, bustling factory. The CEO, representing a hormone or primary messenger, stands outside the building. The CEO never actually steps foot inside. Instead, they shout an order through a window to a floor manager, who represents the cell receptor. The floor manager then blows a loud whistle, instantly alerting thousands of workers inside the building to start the machines. In this analogy, those thousands of workers running around inside the factory are the secondary messengers. They are the ones who actually carry the message deep into the cell and make the physical work happen. Now that we have our factory analogy in mind, let us look at the formal biological definition. In the realm of cell biology, secondary messengers are small, non-protein, water-soluble molecules or ions. Their main job is to relay signals received at receptors on the cell surface to target molecules inside the cytosol or nucleus. When a primary messenger, such as a hormone, binds to a cell surface receptor, it triggers a physical shape change. This activates an intracellular enzyme that rapidly generates massive amounts of these secondary messengers. Because they are incredibly small and highly mobile, they quickly diffuse throughout the entire cell to interact with various proteins. Building on that definition, let us identify some of the specific molecules that act as these internal workers. There are several common and highly studied examples of secondary messengers in human biology. The most famous one is cyclic AMP, which we will look at in detail shortly. Other crucial secondary messengers include calcium ions and inositol triphosphate. Even though they are chemically different, they all share the same fundamental property. They are small, fast, and capable of spreading a signal rapidly throughout the interior of the cell to trigger a biological response. You might wonder why the cell uses this complex relay system instead of just letting the hormone do the work directly. There are two main reasons. First, many primary messengers are large or hydrophilic, meaning they physically cannot cross the protective, fatty plasma membrane of the cell. Secondary messengers beautifully solve this problem by acting as the internal communication network. Second, and perhaps more importantly, this system allows for signal amplification. This amplification is absolutely critical for a rapid and robust biological response to danger or stress, translating a tiny external whisper into a massive internal cellular action. To truly appreciate the power of this amplification, we can look at the mathematics behind it. We can represent the total number of activated final product molecules using a simple multiplicative cascade formula. A total equals R times G times E times M. Let us break down these symbols. A total is the final number of secondary messenger molecules generated. R is the number of receptors activated on the outside. G is the number of G proteins activated by each receptor. E is the number of effector enzymes activated by each G protein. Finally, M is the number of secondary messengers produced by each effector enzyme. If any of these variables increases, the final output grows exponentially. Now that we have our formula, let us see this with a concrete example. Imagine a liver cell responding to the hormone glucagon during a period of fasting. We are given that exactly one glucagon molecule binds to one receptor, so R is 1. This single receptor activates 20 G proteins, making G 20. Each G protein activates exactly one adenylyl cyclase enzyme, so E is 1. Finally, each enzyme rapidly synthesizes 500 molecules of cyclic AMP, meaning M is 500. We substitute these numbers into our cascade equation and multiply them step by step. The final answer is 10,000 molecules of cyclic AMP. A single hormone molecule has generated 10,000 active messengers inside the cell. This massive number of cyclic AMP molecules leads us to an important question. What do they actually do? They trigger a downstream kinase cascade. Let us define the formula for the first step of this phase. The number of active kinase enzymes, P, equals the total cyclic AMP divided by 4. We divide by 4 because each kinase enzyme physically requires exactly 4 cyclic AMP molecules to bind to its regulatory subunits in order to release the active catalytic subunits. Increasing the initial cyclic AMP will directly increase the number of these active kinases. Let us apply this new formula to our previous result to see the cellular effect. We know from our first calculation that we have 10,000 cyclic AMP molecules floating in the cytosol. We need to determine how many protein kinase A enzymes are activated. We take our total pool of 10,000 and divide it by the constant 4, since 4 messengers are needed per kinase. Performing this division gives us 2,500. So we now have 2,500 fully active kinase enzymes ready to do work in the cytosol. Now that we have our active kinases, we can calculate the final cellular response. The total number of active target enzymes, T, equals P multiplied by 100. This is because each activated kinase can phosphorylate and activate 100 target enzymes that break down glycogen into glucose. This formula represents the final massive wave of the biological cascade. If the number of active kinases increases, the final number of target enzymes will grow by a factor of 100, showing the immense power of this secondary messenger system. Now we take those 2,500 active kinase enzymes and move to the final step of our cascade. We multiply this number by 100, which is the number of target enzymes each kinase can phosphorylate. The result of this calculation is 250,000 activated target enzymes. Physically, this means that just one single hormone molecule on the outside of the cell has ultimately turned on 250,000 glucose-producing enzymes inside the cell, providing an immediate and massive surge of energy. Let us visualize everything we have just calculated by looking at this diagram of the amplification cascade. On the left, you can see the cell membrane. Notice the single primary messenger ligand binding to the G protein-coupled receptor on the outside. This is our CEO from the factory analogy. Inside the membrane, the signal transfers to the G protein and then to the adenylyl cyclase enzyme. Look at how the arrows branch out from the adenylyl cyclase, representing the massive production of cyclic AMP from ATP. These cyclic AMP molecules then bind to protein kinase A, shown on the right, which further branches out to activate countless target proteins. This diagram perfectly illustrates how a tiny external trigger cascades into a massive, widespread internal response. To wrap up our lecture, let us review the most important ideas we have covered today. First, Secondary messengers are the vital internal communication network of the cell, acting as the workers that carry out the CEO's orders. Second, they are essential because they take an external signal that cannot cross the membrane and translate it into a widespread internal response. Finally, by utilizing a cascading mathematical amplification system, molecules like cyclic AMP ensure that a tiny initial trigger results in a massive cellular action. You have now mastered the fundamental mechanics of cellular communication. This beautiful biological machinery ensures that our cells can react instantly and powerfully to keep us alive.